Hey guys, welcome to another A-Level Biology video, and this time we will be doing past papers from May-June 2011. To be specific, we will be doing paper 5 and the version is version 1. So, a student investigated the storage tissue in potato tubers of different ages. A newly formed tuber and in an old tuber that was just beginning to form new shoots. So, in one investigation, the water potential of different parts of the tubers were investigated using discs of potato tissue and sucrose solutions of different concentrations. And we have figure 1.1, and it shows the appearance of the tubers and the places where the tissues were removed. And so this is a newly formed tuber and this is the old tuber which has begun developing new shoots. So basically from each of these, parts of tissue were removed for experimentation. So one sample was taken from the inside of the tuber next to a bud, and one was taken from the inside of the center of the tuber. And they're basically the same places for the other one, because tissue is again taken from the inside of the center of the tuber, and tissue is taken from the inside of the tuber next to a new shoot, because it is from the bud that a new shoot emerges. So they're basically the same places. So the results of the investigation are shown in figure 1.2. See, so on the y-axis we have percentage change in mass, and a positive value implies that the mass is increased and a negative value implies a decrease in the mass, and 0% means there wasn't any change in the mass. And on the x-axis we have concentration of sucrose solution, and we've got a number of curves here, 4 to be exact, because we did take 4 distinct samples. So the complete line is for the old tuber, the complete red line. Right? So this is the old tuber, and the complete one is for near the shoot, and the incomplete one is for the central region, and for the blue it's for the newly formed tuber, and the complete one is for the near the bud, and the incomplete one is for near the central region or on the central region. So you have to describe a procedure by which the student could have obtained the results in figure 1.2. So not only do we need to write the experimental procedure, but we have to also detail how exactly we processed the raw data to give the results which we then plotted, because what they're asking for is how they obtained the results that are shown in figure 11.2, and that implies all of the process that finally leads up to us getting that processed data in the form of a graph. So number 1, the student would need a couple of different sucrose concentrations, and at least, because how many plotted points does the student have? 1, 2, 3, 4, 5, 6. So the student needs 6 different concentrations of sucrose solution: 1 molar, 0.8 molar, 0.6 molar, 0.4 molar, 0.2, and 0. So 0 is basically just distilled water. So actually just 5 concentrations, and the student can get that in a number of ways. He can form a stock solution and then dilute and the stock solution to form the other concentrations. For example, the student can make a 1 mole per decimeter cube solution of sucrose, and then he or she might make the different concentrations from that stock solution, or the student might make an initial amount of 1 molar and use serial dilution, but that is unlikely to work properly. So simple dilution it is. So how does the student get the initial 1 mole per decimeter cube solution? Well, to get that, the student needs to dissolve 1 mole of sucrose in 1 decimeter cube water. And what is the mass in gram of 1 mole of sucrose? Well, it is equivalent to the mR of sucrose. And you know the formula of sucrose, it's C12H22O11. So it's fairly easy to get the mR from it. Since the molecular formula is C12H22O11, so the mR will be 12 into 12 plus 11 into 16 plus 12 into 1, and whatever value you're getting is the mass in gram you need to put into 1 decimeter cube of distilled water to make the stock solution. I'm really too lazy to add those numbers up. And if you don't want to make 1 decimeter cube, so if you want to make half the amount, you'll have to half the amount of sucrose in grams that you need to put in. So if it's 500 centimeter cube stock solution that you want, you'll have to use half the mass in gram of sucrose that you would have needed to make 1 decimeter cube of stock solution. And when you have that and you want to make 0.8 mole per decimeter cube, you'll have to take 80% of the liquid portion will have to be the sucrose and 20% would be distilled water, and you'll have the proportions will be the same, and they're depending on how much of the 0.8 mole per decimeter cube solution you want. For example, if you want 100 centimeter cube of 0.8 molar solution, you'll have to take 80% of 100, which is 80, that is 80 centimeter cube of sucrose solution and 20 of distilled. And so basically, number 1, make up 5 different concentrations of sucrose solution, that is 1 mole, 0.8 molar, 0.6, etc. Number 2, make 500 centimeter cube of 1 mole per decimeter cube sucrose solution by dissolving this much mass of dry sucrose in this much water. Number 3, you need to detail how to get the rest of the concentrations using simple dilution. Number 4, you need to mention that the last concentration is actually 0 molar, meaning this is distilled water, right? So it's not exactly a control, but it's sort of a control. And what does the student do? So in each of different concentrations, and because we are actually investigating 4 different parts of the tissue, we'll need 4 volumes of each 4 separate volumes of each, the different concentrations, and we need to place the 4 different types of tissue that we have separately in those volumes, and they have to be in that volume for the same amount of time, and that is for the sake of standardization. But before we can do that, we need to actually measure the mass of the tissue before we put it into the volume, because we want to calculate the percentage change in mass, right? So we'll have to record that, and afterwards, when we have had that placed for a given amount of time, say 2 hours, when we remove it, we'll have to gently mop off any water, any of the solution that is over there. We'll have to be really gentle about it so that we don't harm the tissue, and then we'll have to measure the mass again, right? And then we'll have to take the difference in the masses over the original mass and multiply by 100 to get the percentage change in mass. And this is stuff you probably know. The thing is, you can get extra marks for really simple stuff which you probably didn't write. So when you're saying we have to get separate volumes of the same concentration for the 4 different tissues, you can also specify that we will contain the volumes in what kind of container. It has to be anything that works. Could be a petri dish, could be a small beaker. You'll get 1 easy mark. And because this is simply diffusion osmosis, there are certain things that affect the rate of it. So you'll have to say that we'll have to be really careful when we're cutting those pieces of tissue, because we want them to have approximately the same surface area, and we want all of the containers that we're working with to be in the same area, so they're basically at about the same temperature. And we want the volume of the sucrose concentration to be the same each time, and we'll need to use something to measure that volume out. Could be a measuring cylinder, and even if you really specifically mention not just that you'll have to weigh the before and after mass, but then you'll be using a weighing mass to do it, an electronic balance to do it, that is another easy mark. You may even score a mark for mentioning that you may put something on top of the petri dish to stop any evaporation, and you can get marks for saying that we can repeat these with different tissues from the center of an old tuber, but approximately the same age and of the same type and the same mass, that sort of thing. So you'll have a lot of values. You can cancel out any anomalous values, you can see patterns, and you can get a mean. So this is really easy, but this is, well, not that easy, so to speak, but you can get marks if you get a hold of the stuff they want you to say. And the mark would be for clarifying the level of risk associated with this. How risky is this? I'd say it's a low risk experiment, but what sort of risk do you think you'll face? Well, it's possible that one of the experimenters was allergic to the plant tissue, so wearing gloves would be a good idea, though I don't really see people being allergic to potato tubers, but let's go with it. And sucrose isn't exactly toxic now, is it? Maybe, like a no sugar stuff is bad for you, that kind of a warning. Nah, I'm just kidding, please don't. So that's it. Anything else? Yep, we mentioned repetition, we mentioned reliability, we mentioned all the sort of equipment we'll need. And did I mention what you will have to use to cut those tubers? We could use a scalpel. That is also risky. You'll have to be really careful when using the scalpel. You might accidentally cut yourself if you're not careful enough. So this is it. So if someone asks you for a proper format, number 1, you'll need to give an overview of the independent variable, which would be the different concentrations and what the dependent variable would be, and that would be the change in the mass, and you'll have to say all of the stuff, all of the variables you need to control, such as temperature, surface area, and that makes it easier. And then you can mention all sort of safety precautions and other cautions to make sure that your experiment is reliable. Easy, difference in mass over original mass into 100. And why did the student opt to use change in mass rather than actual mass? Because the thing is, the different tissues are unlikely to have the same mass. You will do your best to ensure that the masses are as similar as possible of different samples, but it's literally impossible to ensure that they are exactly the same. So for the sake of comparison, you use percentage change in mass. And this is the same data once more. And the student looked at the graphs in figure 1.3 and estimated the water potential from where there was no change in mass. The student decided to find out if the difference in the water potential between the central region of the old tuber and the central region of the newly formed tuber was significant. So that was the goal he had in mind. And 20 samples of tissue were taken from the central region of each tuber. So there were two different types of tuber, and 20 samples were taken from the center of each. So in total you had 40 different pieces of tissue, and the change in mass was made separately for each sample using the same procedure as in the original investigation. Why the t-test is a suitable statistical test? Because you can use it to compare the mean of something. Explain how the student should use the value for T to find out if the difference in water potential is significant or not. Well, the student can figure out the degree of freedom, which will be 38 in this case, and from that, for a degree of freedom of 38, he or she can find out what the critical value is, and from that he or she can decide whether or not the tuber is significant. The differences in the water potential of tubers is significant. And from the readings in figure 11.3, the student concluded that the tissue in the old tuber close to the growing shoot has the lowest water potential. So what was concluded is that the region of the old tuber, which was near the new shoot, had the lowest water potential of all others. And what is it? This is the water potential, and it's approximately 0.52. And do you think this has the lowest water potential? Maybe, because when it is placed at the highest concentration of sucrose, there is no net flow, and that implies that the water potential is the same, and this solution is isotonic. And because this has the highest concentration at which this isotonic behavior is achieved, it means it has the lowest water potential, because when solute potential increases, water potential falls. In the old tuber, close to a growing shoot, starch reserves were being converted to sugar. Well, if starch reserves were being converted to sugar, then the water potential should indeed have fallen, because sugar is soluble and therefore affects osmotic potential, whereas starch does not, being insoluble and all. So this is something he or she may hypothesize, but conclusion is a strong word here. In the old tuber central region, starch was being converted to sugar. That was also concluded. Does that make sense? Well, the water potential is fairly low there as well. So maybe, but conclude. And in the newly formed tuber, all the sugar had been converted to starch. This is actually interesting because they do indeed have a high water potential, but there is nothing to suggest that such a a conversion had taken place. Maybe, but not. But can we say that all of them have been converted? We certainly cannot. Evidence which supports these conclusions and the evidence that does not. The fact that the tissue in the old tuber achieves zero net osmosis at the highest, or when in the highest concentration of sucrose, means that of all four it indeed has the lowest water potential, because solute potential is inversely related to water potential, and this should be worth 1 mark. And that is the evidence that supports it. But what about these points? Are there any evidence that support it? Well, the fact that the old tuber central region had a lower water potential means that this hypothesis is reasonable, because sugar is capable of affecting the osmotic potential and is likely to bring the water potential down, but there isn't any evidence for that. That is just a conclusion. And again, here the reason it has such a high water potential might be because all of the sugar existed in the form of starch, but nothing is exactly 100% portraying it. So when we're talking about for the old tuber near the shoot, when we place it in distilled water, it has the highest change in mass, which implies that the most amount of water has entered it, and this actually further supports the first hypothesis that it was at the lowest water potential, and that is why indeed more water should have entered it. And that is the same for all concentrations, and that is 100% in favor of the first hypothesis, because the fact that each of the concentrations from 0 to 0.5, for the same concentration there is apparently a greater change or a greater increase in mass. Say when you're talking about, right, so that indeed supports it, but here's the problem. When we talk about over here, that while this may support the fact that at 0 of all 4 possibilities the most amount of water is entering, and that would imply that the greatest osmotic pressure exists, and the same is for this concentration, for this, for that, for that, for that, for that, for that, until here, that is all right. But when we continue to place it in higher and higher concentrations, obviously water would begin to leave, and that also makes sense. But if we were to compare this, at 0.6, the least amount of water had left for this, and the least amount of water had left here as well, and here it has left. So is this in keeping with the conclusion? Because if this had, or if the potato tuber had the closest concentration to the concentration in which it is put, then indeed less water would have left, but more would have left for this, because there would have been a greater difference, because this would have a higher water potential. So more of this would have left, but less of this would have left, because this had a lower water potential relative to all of these values in the first place. So all of this is an evidence of our conclusion. But we don't really have anything going here. In the old tuber, starch reserves were being converted to sugar. And this would make sense but there seems to be an overlap over here, which doesn't make a lot of sense. So if starch reserves are being converted to glucose, we would expect a lower water potential. But at this point, the old and the new tuber actually has a high water potential, and this is all that we can conclude at the moment. Suggest what further investigations the student could do to provide more support for the conclusion about starch and sugar in the storage tissue. You could carry out the Benedict test, or you could carry out the iodine test, and their color changes will either confirm or reconfirm the conclusions that newly formed tubers do not form shoots until a period of dormancy lasting several months have occurred, taken from tubers of different ages to test the hypothesis. The student found that newly formed tubers do not form shoots until a period of dormancy lasting several months has occurred, and the student investigated tissues taken from tubers of different ages to test the hypothesis. They could test the concentration, and initially they would have been high concentration, and with time and the concentration would have fallen until eventually it was negligible, and finally shoots would start to form. Solutions from 0 to 1. Allow any volumes in correct proportions for making the sucrose solutions. Do not allow if reference to serial dilution unless it would give the correct concentrations stated by the candidate, and ignore reference to 0 as sucrose solution because it is just simple plain old water. So reference to using distilled deionized water, I believe I did mention that, and reference to leaving the plant tissue for a suitable amount of time. I said about 2 hours, allow in terms of long enough for osmotic change to occur. So 3 marks, dependent variable, reference to using tubers from each region in separate containers of each molar solution. Yep, I definitely said that. Look for containers but give if done for one set. Reference to weighing before and after immersion in sucrose solution. Standardization, reference to using the same number, mass, weight or volume of potato. Yep. Known, same volume of each molar solution, same time in solution. Reference to blotting tissue dry before reweighing, suitable method of keeping the temperature constant, and reference standardizing the source of the material. Same size, ignore amount, allow surface area, allow the idea of tissue being fully immersed. You need water bath incubator, temperature controlled room, same species, variety of clearly the same tuber, low risk investigation, cutting away from the hand using tile for cutting, plant allergies, reference to minimum of 3 repeats and a mean in order to spot anomalous data. Yep. Idea of the proportional change from the original mass, allows comparison to be made if the starting masses are not exactly the same. This is a really good way of expressing what I found it a bit hard to do, so I'll write this down. Allows comparisons to be made if the starting masses are not exactly the same. This is actually something or a direction that they often ask about. So it's a good idea that you get a know of how to express your idea clearly. So comparing mean values of two sets of data using a probability table at 5% probability to safety value is higher or lower than the critical value using 38 degrees of freedom. And this is easy. Allow tuber at growing shoot equilibrates at the greatest concentration, so has lowest water potential, greatest change in mass at the smaller concentrations, and that makes sense because sugar does osmotically change, cause a fall, starch would not. But the one thing that invalidates is that while this does suggest a difference in solutes, but not the identity is not clear, and we certainly do not have enough data points to reach a valid conclusion. Appropriate named test for starch, sugar, sucrose. Young tubers would have high concentration of inhibitor, old tubers would have a lower concentration. And this is it. And before this, I'll also read some articles from the internet about osmotic questions, the sort of questions they give regarding this topic. The sum of the original unchanged solution can be seen to fall, rise or disperse when added to a known molar sugar solution. So this is a very interesting thing. And there are also these I found to be pretty interesting. I wonder why they're not opening up yet. Osmosis in bell pepper pericarp tissue. This is a new version of the classical osmosis practical, measuring the weight change of tissues in various osmotic solutions using pieces of bell pepper instead of potato. This is a reliable way for students to plot and use calibration curves in order to eventually work out the osmotic potential of the pepper. The giant cells are seen on the inside of a pepper when sliced open and can be popped with a mounted needle showing the cell contents and indeed under pressure. The cells of the endocarp have thick wavy cell walls which allow for expansion. Further questions and a suggested investigation with whisking tubing prompt thinking about turgor pressure in plants and why it is important. So this is it. Measuring the water potential of a potato cell. You need two balances that are accurate to 0.1 gram. You need potato tissue, you need marker pens, you need kitchen knives, and you need graph paper, and you need pipettes, and you need test tubes, and you need a test tube rack, and you need beakers, and you need a test tube filled with 1 molar sucrose, and you need distilled water, and all the beakers, the test tubes, and the stock solution plus distilled water are for you to make the different considerations. Methylene blue solution in test tube and Pasteur pipette. So instructions: prepare 10 centimeter cube each of 0.8 molar, 0.4 molar, 0.2 molar, 0.15 molar and 0.075 molar sucrose solutions each in a separate tube. Be careful to mix the water and the more dense sugar solution thoroughly. Add a 6th tube of water to complete the series. Label each solution and another tube to match. Example, 2 tubes labeled 0.8 molar and 2 labeled 0.4. Cut up about 10 gram potato tissue into small cubes about 2 millimeter square. Weigh accurately about 0.75 gram of potato tissue into each empty tube. Load the tissue before weighing and be sure that the balance is carefully zeroed, and record the weight in each tube accurately. When all the weighings are complete, cover the tissue in each tube with the corresponding solution. 5 centimeter cube is sufficient, and note the time. That is, cover the tissue in the 0.2 molar tube with 0.2 molar sucrose. Note how the potato looks when this is complete, and leave the tubes for 15 minutes, swirling occasionally, as in moving the test tubes, and you have enough time to make a diagram of the technique so far. And then you add 1 drop of methylene blue solution to each tube with potato tissue in, and mix it with the contents of the tube. And you need to arrange your eye levels with the tubes, and the pipette carefully withdraw some of the colored solution from each tissue tube in turn and add it very carefully to the corresponding sucrose solution tube by immersing the pipette to about half the depth of the solution and gently expelling one drop. So you have to remove some blue solution from the 0.8 molar solution with the potato tissue in it and add it to the 0.8 molar stock tube that is completely unchanged. And you need to wash the drop carefully and record your observations. In some it won't move, actually that won't happen. In some it will move upwards and some it will move downwards, and you need to also record the time it takes for it to move a certain distance. Remove the tissue from each tube, blot it on a paper towel and weigh each amount separately, making sure that the balance is carefully zeroed before each weighing. Make a table of initial weight, and final weight, initial weight for each concentration, and plot a graph of final weight over initial weight, which is actually the ratio against the concentration. And then using your graph, you'll have to predict the concentration of sucrose, which is in equilibrium with the potato tissue, and hence calculate the water potential by consulting the table at the foot. I'll just leave this. So if you have to do this by weighing, it will be much easier. We just did that one. Prepare 10 centimeter cube of each of the 0.8, each in a separate tube. Be careful to mix the water. Same instruction, labeling. You have to cut about 10 gram and you have to have about 0.75 in each. Blot the tissue before weighing and be sure the balance is zeroed. Record the weight in each tube accurately with corresponding solution. Leave for 15 minutes, remove the tissue, blot it on a paper towel and weigh each amount separately. And you need to make sure that the balance is carefully zeroed before each weighing. Make a table of initial weight, final weight and final weight, initial weight for each concentration and plot a graph, and using that you can make your predictions. So this one actually has both of these. And for the first method, explain the behavior of the drops in part 7, how that method can be used to measure the water potential, because of one for which there would be no movement. That means there wasn't any movement of water for that test tube and that potato. So that was the water potential of the potato. Note any errors in this method and think especially of what happened to the blue drop in the pure water tube. What would happen? Obviously it would be much heavier, so it would go downwards very quickly. Or are we talking about the potato that was in the water? In that scenario, there would be a lot of things that would leave, or something of the sort. So if we were to place potato in pure water, a lot of water would enter the potato tuber and a lot of things would leave. So the concentration would basically undergo some changes. And that's it. Hopefully this was of help to you. And this is a similar experiment, not quite the same, but has to do with osmosis, the bell carp one, observing giant cells. So which one should we start with? Technical and teaching. They have to determine the water potential of cells in the pericarp by investigating the change in mass that is produced when water moves into or out of the cells by osmosis down a gradient. Mass are easily detected using 2 decimal place top pan balances, 1 molar sucrose solution, so plants contain toxins, so allergies are a concern. Sharp instruments, and this is what you need. You need half of red pepper, 50 centimeter cube of stock solution, 50 centimeter cube of distilled water. You need 11 test tubes, graduated pipettes, a pipette filler, cutting tile, kitchen knife or scalpel, petri dish with a lid, paper towels, graph paper and blunt forceps. So modeling changes in cell agar and suggested apparatus: whisking tubing that have been softened by soaking it in distilled water for 5 minutes, 1 molar sucrose solution, boiling tubes, distilled water, boiling tube racks and thread. Possible method for modeling changes: securely knot the piece of whisking tubing about 1 centimeter from the end to make a whisking tubing bag. A simulation of the cell: fill 2/3 of the whisking tubing with the 1 molar sucrose solution, which is basically simulating the cell sap in the vacuole. You need to carefully squash out the air from the space above the sucrose solution in the whisking tubing bag. Then securely tie the thread around the top of the bag to close it. You need to place a whisking tubing bag which is acting as a model cell into the boiling tube and fill the boiling tube with distilled water. The distilled water is representing the water molecules that are able to diffuse into the model cell from surrounding cells. You need to make a drawing of the whisking tubing, and show its appearance it would basically be really bloated, really swollen. And you need to place the boiling tube in the boiling rack for 30 minutes. And after 30 minutes, you need to describe the appearance and make a drawing that will allow you to compare its initial appearance and its final appearance. Expected results: water will have diffused by osmosis from the boiling tube through the whisking tube into the 1 molar sucrose solution down a gradient. This is the same thing that happens with a potato, because water will either enter or leave, nothing else can enter or leave. So if you place it in distilled water, a lot of the water would have entered the potato, so the water potential of the water would have fallen, because now there would have been a lot of solute, because none of the solute would have left with it. The volume of liquid inside the bag would have increased, and so the pressure would have increased, and this increased pressure is simulating turgor pressure, and the model cell is now much more rigid. Changes in cell turgor: slow or fast. So pressure changes inside your whisking tubing take place slowly. And how could you speed it up? You could increase the temperature of the model system, or increase the steepness of the water potential gradient on either side of the semi-permeable membrane. Why are the pepper pieces stored in a covered dish until they are needed? To reduce the amount of water that may evaporate from the cells. Without this, those pieces cut first could have a lower starting water potential than those cut later. This is important. They could give you something of the sort. They might ask you why they put a particular specimen of cells whose water potential you were investigating in water before using it. This will be your answer then. Why are the discs washed with distilled water? To remove any solute molecules that may have been released from the cells when the tissue was cut. If not removed, these solutes could change the water potential of the solutions that the pepper pieces are being placed into. Again important. Why are they dried very carefully? To carefully remove any and all surface water without rupturing any of the cells. Explain why the pieces are placed for 30 minutes. After 30 minutes, what happens to the net diffusion of water molecules? There won't be any more net diffusion. To allow water molecules to diffuse by osmosis between the bathing solution and the cells of the pepper tissue down a gradient until water equilibrium is reached. At equilibrium, there is no further net movement of water into or out of the cell in the pepper pieces because the water potential on either side is now the same. And why are they dried before being reweighed? To remove surface water, which would contribute to the final mass of the pepper but would not represent water that has been taken up by osmosis. Why was data converted to percentage change? Because initial starting masses are variable and this conversion makes comparisons possible. So use a graph to find which molarity of sucrose solution has the same water potential as the pepper pericarp. Explain. The molarity of sucrose solution with the same water potential as the pepper will produce a percentage change in mass of 0%, for there will be no net loss or net uptake. And how do you find the molarity? Finding the intercept of the line on the graph with the x-axis allows the molarity to be calculated. Explain fully the shape of the graph in terms of water potential. That is why some pieces of pepper pericarp tissue gain mass, why some lose, and why some remain the same. Obviously because of differences. Explain how you obtain a value for the water potential by creating a calibration graph from the data provided and reading off from the graph. How do you investigate the reliability? Set up replicates at each sucrose molarity and calculate a mean, percentage change, and pool class data because you compare it. Compare the value for water potential of pericarp cells that you obtained with that by the students. Discuss the possible causes of similarity and differences, variation in water potential of pericarp cells within a pepper, between different peppers, human error, inaccurate pipetting, inaccurate use of balance, inaccurate record keeping. So, practice.